Okay, now we're going to look at some biological and anatomical differences between modern humans and Neanderthals. Neanderthals and humans differ in both their mitochondrial DNA and nuclear DNA. There are also many visible morphological differences, though these differences appear as gradients within types without finite cutoffs. So we're seeing a range of looks here, and there is a great museum called the Neanderthal Museum. It's in Germany, and if you go to their website, they do something fun where they reconstruct what Neanderthals would have looked like, based on the skeletal evidence, and put them into flesh or sculptures, recreate them. And then they put modern clothing on them. And you can kind of see that, Neanderthals, they do look different, but if you put them on the street with a hat and a trench coat, they could pass for, anatomically modern humans. So they were not so different from us, but we can see some, general features that are different. So here on the graph or the graphic, you can see a list of things that are different. A lower, larger cranium, larger brow ridge, larger nose, larger shoulder joint, larger, broader rib cage. You can see that the rib cage kind of flares out at the bottom. Larger elbow joint, broader hips, shorter forearm, larger hip joint, larger and thicker kneecap, shorter and more flattened tibia, and a larger ankle joint. In general, Neanderthals show many differences in cranial morphology, so that morphology of the head and skull, from anatomically modern humans. And we've talked a little bit about these earlier, but features include large arching brow ridges, no chin, so no bony protrusion from the bottom of the mandible, a pronounced occipital bone in the back of the skull, low foreheads. So we're going to look at this next slide. Okay, so in this graph, the top two skulls are Neanderthal, and the lower two skulls are anatomically modern human. So we're seeing the Shanidar and the Laferzi, the two classic Neanderthal finds, and you can compare the two at the top and look at their similar features: long, low brain case, occipital bun, large, wide nose, no chin, which is an interesting thing. A large gap behind the third molar, which humans do not have. We have smaller dentition and it seems smaller jaws. And we don't have that extra space behind the third molar. So when we get down to the lower two skulls, we're seeing a rounder, higher brain case, all right? So just compare the kind of football-shaped skulls of the Neanderthals to the rounder shape of the skulls below, the anatomically modern humans. And there's a forehead here. So kind of imagine if you thought these people might have a fever, trying to put your hand on the forehead and take a temperature. We definitely are seeing some foreheads in the Quaffse and the Piedmosti skulls on the bottom of this graph here. We're seeing the chin or bony protuberance at the bottom of the mandible, smaller nose, a flatter mid-face, and a more rounded occipital region rather than that bun shape in the back of the skull on the Neanderthals. Okay, we have moved on from Neanderthals. The next species to cover are Denisovans. So they're kind of a mystery. In 2000, 2008, and 2010, researchers found fragmentary hominin remains in Denisova Cave in southern Siberia. Only a finger bone and two teeth were found, and these are dated to as old as around 80 to 60,000 years ago. And as it turns out, the DNA evidence from the finger bone does not match that of either a modern Homo sapiens or Neanderthal. Within 2 years, its entire genome was sequenced, and the data confirms that the Denisovans were an entirely separate lineage. There'll be a little bit more information on Denisovans and their DNA in this week's video. So as more time passes, we're finding more DNA clues and DNA evidence. In the past few years, exciting results have been announced about DNA found in more than a dozen different Neanderthal fossils dated between 50,000 years ago and 32,000 years ago. DNA evidence suggests that other fossils from Central Asia, dated at 48 to 30,000 years ago, are also Neanderthals, or an entirely different species. The technique used involves extracting mitochondrial DNA, amplifying it through PCR, and sequencing nucleotides in part of the molecule. Major advances in molecular biology have allowed much more of the Neanderthal genetic pattern to be determined, with the ability to now sequence the entire mitochondrial DNA sequence in several individuals, as well as big chunks of the nuclear DNA, making it possible to map the entire European Neanderthal genome. And it has been found that Neanderthal DNA is remarkably similar to modern human DNA, with 99.84% of it being identical. Researchers also sampled individuals from various parts of the Old World and concluded that many people today still have Neanderthal genes, found in all except African populations. It's believed that shortly after Homo sapiens migrated out of Africa, a few interbred with Neanderthals before they dispersed to other areas of the world. The degree of interbreeding appears to be small but significant, with 1 to 4%, kind of evidence in the DNA for living non-Africans. Next, we're moving on. Next species, the little people, Homo floresiensis. And I don't have any grand conclusions from this part of the lesson, but I'm going to show you kind of what was found and the information we have. So, we're skipping ahead a little bit, but by 25,000 years ago, anatomically modern humans had evolved and dispersed throughout the Old World, but there may have been a late surviving population of erectus-like humans on the island of Flores, Indonesia. The remains of 9 individuals, dated to as late as 13,000 years ago, were discovered in the Liang Boa cave in 2004. The more complete female adult skeleton, LB1, is barely 3 feet tall, with a cranial capacity of only 417 centimeters. Their unique morphology of just being extremely tiny people earned them a new species designation, Homo floresiensis. And here's an example of what the skull would look like, or it is the fossil found. Recently discovered stone tools were radiometrically dated to at least 1 million years ago, suggesting that a population, potentially Homo floresiensis, had been living there for a long time. Some have just suggested that this individual actually has a disease called microcephaly, or a condition called microcephaly, so just a very small person. So we're looking at hypotheses as to why these people are so small and who are they. Others, such as UC Berkeley anthropologist Gary Richards, argues that the Flores individuals are not an outlier or example of people with a disease or condition, but have unusually small body and brain sizes due to microevolutionary adaptations to their island habitat. This is also known as island dwarfing or insular dwarfing, and has been observed in other mammals, including the dwarf elephant. So basically, in mammals, sometimes when the environment is confined, it is more adaptive to have a smaller body size, so people, or whatever species it is, evolves to be smaller. So that brings us to the end of our lesson on archaic humans. You do have a video this week if you're taking my physical or biological anthropology class. We will be reviewing the TED Talk, Savante Pabo, DNA Clues to Our Inner Neanderthal. This concludes our archaic humans lesson. Thank you for listening.